Hi, I'm Jennifer from OpenHelix. Welcome to this tip of the week. If you're viewing this movie somewhere other than on our blog, be sure to check out the full post at blog.openhelix.com to be able to see the full discussion with links and references. In today's tip, I'm going to introduce you to a resource that I found through the Nucleic Acids Research Database issue from this year. And the resource's name is VnD, which you can see from the article's title is a structure-centric database of disease-related SNPs and drugs. Now let's head over to the VnD homepage, which is where we're at. You can access it at the address shown or from the links in the blog post. What attracted me to this resource from the NAR article was this figure, which you can access on their site from the Methods and Guides tab. And it shows that the VnD compiles an intriguing number of databases information. A lot of these databases OpenHelix knows, respects, and even trains on. And then, in addition to the information that they collect from resources such as Uniprot, RefSeq, OMIM, PDB, Drug Bank, etc., they also do an analysis of their own, comparing the wild-type versus mutated proteins. They do structure modeling, predictions, as well as docking with ligand predictions, and then they provide an analysis of the protein structure and drug docking compared in wild type and the mutant versions of the protein. So you can see how it might affect disease, drug, ligand binding, etc. So for a quick example, we're going to go back to the VnD homepage. Here's a paragraph describing VnD. I'm not going to go through that. You can look at it on your own. There are also areas for search. You can see you can search by keyword, a variety of different formats, or by sequence and structure analysis. In today's quick tip, I'm going to use their RS example. As you probably are familiar with, RS numbers are from NCBI's DBSNP database. So here is our results report. We get SNP information, so a link to DBSNP. It's on chromosome 19, the position, the strand, the allele change. It shows you the gene that the variation occurs in and gives you links to a flanking sequence. Under that are an analysis of the actual functional and structural effects. A link to RefSeq. The change occurs in exon 7 of the gene. It shows you various protein information, including the residue change. You can link to Uniprot information, and this image here and this logo links you to the VnD analysis. Here we are looking at the VnD report. It shows you the disease terms that may be associated with that structural or with that change. Then you get a chart of variation structure analysis. It gives you various general information about the protein, as well as then the structural changes that each variation occurs. So they show you the variation change. They also show you the properties of this area of the protein before the change and after the change. They show you the pocket that the change occurs in. And if you click this structure viewer image, you're taken to the VnD's protein structure visualization report, which includes a lot of information, including a JMOL viewer, where you can examine the differences in the changes between wild-type viewer and mutant viewer. And this viewer has all the normal JMOL functionality, which we teach in other tutorials. Thanks!